So welcome, Dr. Geis. We have our special guest today. Phil Geis is a microbiologist. So what is a microbiologist after all?
Well, a microbiologist is somebody that studies small stuff, bacteria, fungi, yeast, molds, viruses, algae, stuff that you can see through a microscope, classically. I've been doing it for an awful long time now. I think I first got interested in microbiology when I read The Microbe Hunters. Did you ever read that by Paul DeCreux? No? He talked about Spallanzani and Pasteur and Koch and Theobald Smith and all these guys that had discovered these interesting microbiological, not just problems. They didn't just solve problems, they solved basic understanding. And Pasteur showed that spontaneous generation wasn't occurring anymore and that kind of thing. That inspired me, and I worked in a high school lab taking mud samples in the Gulf of Mexico for looking for actinomycetes. I went to the University of Texas and studied microbiology there. And then I was in the Army for 3 years doing microbiology, clinical microbiology, and still loving it. I went back to graduate school. I got a PhD in mycology and microbiology. I like mycology almost as much as I like microbiology. I like fungi.
Mycology?
It's a study of fungi, like yeast, molds, Aspergillus, Penicillium, Saccharomyces. Then I went to work for Procter and Gamble in Cincinnati, Ohio, and that was the southernmost place I could get a job. I'm a native Texan, but it wasn't the Northeast. I didn't want to go to the Northeast. It's too cold. So I worked at Procter and Gamble for 30 years, mostly as a microbiologist or managing microbiology and other scientists, and I still liked it. I got hooked up with editing journals in microbiology and representing the company in various organizations as a microbiologist. I retired back in 2011 and started a consulting group in affiliation with the Advanced Testing Lab out of Cincinnati, and I'm still doing micro. As we were discussing, I was doing micro on the way over here today. So I like microbiology a lot, Monica.
It's a fun subject.
It is.
It's really great. So we call our class Microbes Without Borders, and you have traveled around the world to solve microbiological problems. Can you give us a few examples?
Oh, sure. Let's see. A lot of my problems that I've solved, the things I've done, have been associated with some kind of spoilage, some kind of biodeterioration or biodegradation. And a lot of times in the context of how a product is preserved, maybe a cosmetic or a drug. And one of the most fascinating ones I ever had nothing to do with really microbiology in general. We were making a drug product in Japan, and it had bacteria in it. It was contaminated. We couldn't figure out how it was being contaminated until we finally looked one night in a vat, one of my colleagues, and saw a very small man with a mop and bucket cleaning out the tank, and just with a mop and bucket. And this was supposed to be a cosmetic drug that was not supposed to have any bacteria at all. So you can imagine somebody tracking around in their boots with a bucket and mop. And that was very interesting.
That's great. There was another good example a couple of years ago in a newspaper with the contaminated medication, the painkiller. Can you address that quickly?
This is the
compounding company.
Yeah, the compounding company. This was a sad story. These were compounding companies, and their job is they're sort of a pharmacy operation. Their job is to prepare final dosages of approved medicines, and many of these guys had gone beyond their original charter and started using aqueous products, and they weren't prepared, much as I discussed with the guy in the tank, to do it right, to do it in a way that would keep bacteria and fungi out. So they compounded these things and they contaminated them with Aspergillus niger and some other fungi. And what the product they contaminated was a steroid that was injected typically into the spinal column, into the CNS, the cerebrospinal fluid. And you can imagine what happens when you inject living fungi at high concentrations into your spine. And some people had very serious infections from it, and the FDA is still acting against these guys, some of whom will probably go to jail.
So you can see there's lots of examples of microbiology, and microbes are clearly without borders. Thank you very much.
Yes, Monica.
And we'll talk about the caves of Glasgow next.
Okay.
So my first question for you is, you have a lot of experience in biodegradation and how microbes contribute to that. What sort of a general vision of what microbes actually do and how do they work in biodegradation?
Well, typically these are cultural properties. They're things that are very valuable to people. Books, monuments, paintings, and they typically are removed from their natural environment, especially in the case of the Glasgow Cave, where they're moved to environments where they're concentrated, and oftentimes in museums where they bring in various microbial flora. And they share that flora just by virtue of air currents and stuff. And if the environment isn't carefully controlled, the people coming through, the food items, the debris from people, and their bacteria and fungi expose the objects to a lot of contaminants. Contaminants of different capabilities. Contaminants that can grow at very low water activities, where there's very little water, grow at relatively low temperatures. And they begin to grow, and there's lots of time. The museums are there for years and decades and eons. And so they grow slowly, but they grow, as we say in the trade, microbes never sleep, and these are things that go right in the night. They never stop.
So what do you think the financial impact is of biodegradation in the world?
Oh, it's profound. And some of the treasures, I can give you an example. It's a little bit obscure. I have a good friend in Germany who's an expert in cultural properties. And he's responsible for some of the major cultural properties in Germany and Austria. He and his wife are divorced because, not that they are unfriendly or don't live together anymore, but because of the ultimate liability that he has. He will lose everything if any of his cultural properties go south. So he has no possessions at all. He's penniless. He lives at his wife's pleasure. But these cultural properties are just simply priceless. The David and Florence, or the Rape of the Sabine Women that a good friend of mine takes care of. All these things, if they're lost, they're priceless. And it's hard to understand the impact on not only our world, but also the individual that's responsible for it. Another element is a political element. The Lascaux Cave in France is also a UNESCO site. And if France doesn't do an adequate job taking care of it, UNESCO has the treaty power to take control of that site, to occupy that cave, and do what they want to with it, which of course would be tantamount to saying UNESCO is going to take over the Grand Canyon in the United States. It's a matter of cultural and national pride.
Right. So the caves have been closed for a long time.
Yeah.
Why were they closed to start with?
Well, they were closed to start with when many decades ago, when they noticed the buildup of what appeared to be algae on the surfaces, somewhat bacteria too. They had brought in floodlights so people could have their lunch down there, and the local folks or whoever were the visitors could enjoy the sights. So they had a lot of floodlights. They had brought in a lot of moisture and food, and they noticed a lot of green patina on some of the cave surfaces that were building up. They tried a number of different things that we can talk about later, but they eventually realized they had to stop people coming in. Initially they said, you can't eat anymore in here or drink. You can't have your lunch down here. And then they realized, well, this doesn't seem to be working. It still deteriorates. So they banned folks entirely.
You think that was the right decision?
Yeah, I think it was. It was the right decision for the paintings, for the caves. I'm sure it would have been nice if folks could have seen it, but there would be no paintings left by this time, probably.
So you've been a consultant for the caves for a while now. What is the actual problem? Why is there still an issue about it?
Well, the issue is that they continue to have breaks down in some of the elements that control the environment inside the cave. The major breakdown was when they changed the mouth of the cave to put in new air conditioning units, because they try to have it somewhat cool and very dry. And in doing so, they changed the entrance to the cave so much so that it flooded. And that caused a lot of problems. And when that happens, they often take the course of applying biocides. And the biocides are water-based, and that usually introduces even more water to the cave drawings. And they pay especially attention to that. So in the end, it's a matter of exposure to again, the things that people are introducing.
So do you think there's a chance that the caves can be reopened, or
not Lascaux?
No, not Lascaux.
Altamira, yes. The government wants to reopen it because it's a tourist attraction.
Right.
And Sayas Aman, the guy that is the leading scientist in this area, says he recommends against it. But Lascaux has become almost a holy site in France. There's a Lascaux group that lives at the cave, and they consider it almost a holy relic site. And they're very sensitive to any comment that people would open it up again. I'm sure they would picket it.
Did you get a chance to get into the cave?
No, I didn't. That's my major regret. I didn't get in the cave.
That's great. So they're building artificial restructures of the caves? Is that what they're doing for the tourists?
Mm-hmm. Right now there is one, a relatively small one that's adjacent to the cave, and it's been painted with reproductions of what's in the natural cave, what's in the original cave. Cool. Yeah, it is.
That's very exciting.
And they're using that for experiments too. It's not a very good surrogate, but it's the best they have.
What do you think is the future of cave paintings, art, and how can we control or how can we keep it protected besides just closing these sites to tourists? Well,
a lot of that stuff now is in the hands of almost artisans. The science on this is not real good, and we can talk about that more in terms of Lascaux in a bit, but this is really in the hands of people who have special techniques. One of the things that the class is reading mentions a fellow named Piero Tiano. And Piero, I met Piero. He is responsible for the David in Florence, the rape of the Sabine women, and some other cultural properties. And he tells he has a biology degree, but he's worked in the field for almost 30 years now. And he's learned as sort of as an apprentice and then as a journeyman the art of cultural preservation. And there's secrets that he won't reveal. He knows how to save this statue. He knows, and he's responsible for the statue day to day. If anything goes wrong with the David, it's his responsibility. And he lives with the statue, but he has special techniques that nobody knows.
That's pretty crazy. So what's the science behind it?
The science is, I think if your students would look at some of the science carefully, and you might even look at Sayas Jimenez, the one that you've assigned, the science is weak. The science at Lescaut is very weak. I can just give you an example. He came to the conclusion that the disinfectants were causing the fungal growth because he imagined a metabolite to the disinfectant, and the fungus grew on that.
Wow.
And it doesn't tie back well. It didn't validate that. So a lot of the science is not published in what you'd call heavily peer-reviewed journals. The peer reviewers are typical conservatories or somebody in the environmental area, without folks that are doing the hard review that you expect in science in some of the molecular journals.
Are some of the interest groups detrimental to their understanding of the science, or
yeah, they are somewhat. Yeah,
polarized?
The group, for instance, Lescaut, that monitors the cave itself, that live there, they don't really want science so much to understand how to prevent the destruction of the cave. They just want science to talk about the cave. So the work produced a fungus that was unknown before, and they named it after the cave. And they thought that was a major victory. It had nothing to do with saving the cave, but it did give it notoriety and a special place in the biological history.
Right. So what's the best way to preserve it besides keeping people out there? Are there any methods, techniques? What we could use?
The basic methods are dryness and cool. And that's almost true almost every place, whether it's cultural properties, parchment and museums, or the cave itself. The drier one keeps it. The less apt you have to have the fungus growing. Typically, it's fungus at low AWs. There is a qualification in that, because the drier you make it, the more water you might pull in from the outside environment. So that it is a problematic solution for some applications, but in most museums it's dry and cool, and the more they can maintain that day to day, moment to moment, the better chance they have of maintaining their properties.
So if our students are interested to learn more about biodegradation and microbiology in that area, what would you recommend for them to do?
I'd look at some of the journals and some of the books. There are books on biodegradation. There are journals the one I edited is called International Biodeterioration and Biodegradation. There are even conservatory schools where one learns to conserve these cultural properties and learns these special secrets. And the Smithsonian actually has internships in conserving cultural properties.
Oh, that's interesting. We need to provide the information to the students. Are there any benefits to biodegradation? Can you see if anything that's valuable, rather than just talking about the negative sides of what microbes do with all these properties? Is there any positive thing that can come out of it?
Oh, sure. Yeah, certainly landfills, we hope there's some biodegradation, not too rapidly. Over time, we expect that to happen. We wouldn't want too many things piling up around the world.
That's true.
Plastic is very difficult to degrade. It slowly degrades with a combination of chemistry and light and microbes. And the chemicals that we use in our daily life, the consumer products, all have some expectation that they'll biodegrade, especially the green cleaners. That their chemicals are designed to degrade relatively rapidly.
Right. Can you make any comments on biodegradation and the oil spill?
And the oil spill, yeah, I can.
They've made some attempts to clean up the oil spills with microbes as well.
Yeah, they have. The record there, as you probably know, has been mixed. I haven't worked in that field for a number of years, but I know at the time I was involved with that, and I did work with the Bureau of Land Management on this, applying special microbes in which we have inserted genetic elements that would theoretically have enzymatic breakdown of oils weren't so successful because the bug didn't survive well. It couldn't compete well. Some folks had a lot of results with just fertilizing the whole oil spill, putting nitrogen sources and things of that nature. So the incident bugs, the ones that are there already, could adapt and try to grow and be encouraged. Some parts stick around forever. As you know, some of the asphaltenes are so heavily condensed with aromatic structures that they're very slow, if almost to degrade almost on a geologic time scale.
Right. So how long does it take to, for example, degrade a small area? What's a time frame of biodegradation?
I mean, in the context of oil?
Can you watch it? Yeah, oil, buildings.
You can see that. I think it's easiest in a chemical sense when you can monitor the change. If you have an oil spill, say it's crude, the short and medium change alkanes go very, very rapidly just by beta oxidation. It just peels off a couple of carbons at a time. Some of the smaller aromatics, like the catechol, the ring opening is fairly rapid, and they become effectively alkanes and can be degraded. But the real highly condensed ones, all the aromatic rings are much more slowly degraded, and the more condensed they are, the more aromatic rings they have, the slower they go. And those are the ones you see as tar balls that wash up on the beach sometimes.
You mentioned a few minutes ago about using bioengineered microbes with different genes in it to have better properties and function. What do you think the perception of the public is to release GMO modified plants, organisms in the environment? I think
it's the public in the United States is not so aware of it. There are some groups that are more concerned than others. I think in Europe there's a much higher concern for that. You see that in some of the restrictions on GMO foods, especially the raising of GMO crops. As far as releasing bugs that have special capabilities like that, I think that's kind of obscure to most of the public. The EPA is responsible for that, and oftentimes it's a special interest group that compels their action on that brings up an issue perhaps of unaddressed pathogenicity or unaddressed impact on the environment.
Do you think that's a feasible solution, or do you think we should be careful with these?
I think we should be very careful. Not so much because I see a present danger, but I don't always see the benefit. I think that there's a lot of in vitro data or pilot plant data that says it works, but doing it in nature is more costly, and the measures are more difficult, and oftentimes we don't have that conclusive data that's going to work in nature.
Yeah, that's true. So do you have any other comments on biodegradation, any suggestion for the students, anything that's really interesting? What was like something that was really interesting to you, what you didn't know before you worked with the caves?
Let me think a second on that. I think for the students, I would encourage them to consider just getting their feet wet on it. Even I don't have any doubts that your museum here on campus or the museum in Gainesville or any of these places has biodegradation problems. It's very slowly degrading. It acquaints you with a different group of microorganisms, something that you don't normally see. I know when I first got involved in the field, I worked with the Museum of Art in Cincinnati, and some of the dresses appeared to have a bit of mold on them. And we sampled them all, and we couldn't find anything to grow. And looking back at that now, I understand fully that I used the wrong media. I just didn't pursue something that would grow at extremely low water activity. And I encourage the students to consider that there's a range of bugs, and it's not just the ones you see in the lab that are so easily cultured.
True. Any other comments on microbiology in the world of microbes and microbes without borders?
I think it's a lot of fun. I think that working overseas, working with Lascaux, working with Procter and Gamble in my previous life in different labs, acquaints you with microbiologists around the world, and their interests and the perspective they have on the micro world and what they're working with. And that's so enlightening and so much fun.
Great. Well, thank you very much for your time today. Appreciate it.
Thank you.
And I think our students enjoyed as well.
Thank you.